I'm Dr. Eran Eyal. I work at the Bioinformatics Unit of the Cancer Research Center at the Sheba Medical Center, the largest medical institute in Israel. In this practice session, we are going to address the emerging topic of personalized medicine in cancer, namely interpretation of individual mutations in tumor genomes. In order to check the significance of a particular somatic mutation, which typically discovered in sequencing experiment of a cancer patient, we'll need to evaluate the mutation, which was found in the cancer cells using several public resources which are available for us. So let's examine, for example, the mutation BRAF V600D, which was discovered in a meaningful frequency in the sequence of the tumor sample of our girl, a patient diagnosed with tongue hemangiopericytoma. This girl was mentioned in the lecture of Dr. Elad before. The first question which we would like to ask here is if indeed the BRAF gene is a cancer-related gene. Sometimes this is a very obvious question if the gene was discovered in a cancer panel, but sometimes this is not an obvious question, like if the gene was discovered in a whole exome experiment. So to answer this question, we will turn to a resource maintained by the Sanger Institute in England, which includes the most comprehensive list of cancer-related genes, and by 2019 includes more than 700 genes. So let's open the Census Gene website. You see from the Cosmic website to the Census Gene, and we are now in the front page of the Census Gene website. Let's scroll down a little bit, and we'll see the list of all genes in Census, and we can search in the search box for BRAF, BRAF, and indeed we see that BRAF is included in the list, so we can be sure that this is really related to cancer. In order to see in which hallmark cancer feature the BRAF gene is associated, we can press now on the hallmark wheel and see which sections are marked. We can see that BRAF, which is a kinase and a component of the RAS-ERK signaling pathway, is involved in functions that provide proliferative signaling and means to escape cell death. We can also see that this gene is an oncogene, meaning that its perturbed function is associated with cancer development. It is therefore expected that its activity is increased in cancer cells, and that cancer-associated mutations in these genes are of gain-of-function nature, often of dominant nature, acquired by somatic mutations in only one copy of the gene. Indeed, we can see that mutations in this gene are reported in general as somatic. Let's go back to the table. It is expected that activity of BRAF is increased in cancer cells, and that cancer-associated mutations in these genes are of gain-of-function nature, often of dominant nature, acquired by somatic mutations in only one copy of the gene. Indeed, we can see that mutations in these genes are reported in general as somatic. In the tumor types column, we can see all cancer types which have been associated with this gene. Interestingly, tongue hemangiopericytoma does not appear, but a variety of other cancer types do appear. Note that the search for BRAF brought additional hits, like the ACAP gene. Most of these genes were found to form fusion gene with BRAF in various cancers, and were retrieved in the search because BRAF is mentioned in their description. We will now investigate the specific mutation which was discovered in our patient, BRAF V600D. Let's open the Cosmic site, which is a comprehensive collection of somatic mutation in cancer, also maintained by the Sanger Institute in Cambridge. Let's search the BRAF in the search box. We'll now press the gene hit which we got. We can clearly see that position 600 is a hotspot for somatic mutations in this gene, with strong peak of mutation events in this position, which is located within the protein kinase domain. Mutations in this position can be either simple substitutions or complex mutations. The narrow distribution of somatic mutations in hotspot regions is very characteristic to oncogenes, as only a very limited set of positions and events can improve the function of a protein. Most mutations event, not surprisingly, reduce efficiency of the protein, which were carefully optimized by a million years of evolution. We can also see the various types of mutations detected in this region, including substitutions, complex mutations, and insertions. Detections of different types of somatic events in the same region is considered another proof that the variant is indeed functional. Let's examine the types of somatic variants observed in this gene. I will click on Variants tab on the left. Change the default value to show 50 variants and sort the variants by decreasing count. In our example, most frequent event type by far are of type V600E, which has been reported in over 28,000 samples. The mutation detected in our girl, V600D, affect the same amino acid position, but is far less common. It is indexed as COS477 and was reported only 26 times in the database. By clicking on the mutation details in the table, we get additional data regarding this particular mutation, including listing of all samples and distribution of the events according to cancer type. Most of the patients who had this exact type of variant suffered from skin cancers, but several had various other types of cancers, and in some cases the exact cancer type was not specified. So we are now familiar with some resources in which we can get information on cancer-related genes and functional variants in these genes, which can induce or suppress the development of cancer. But what can we practically do with this knowledge in order to benefit the patients? There are some resources which get a bit closer to clinical applications. One of those is My Cancer Genome, which is maintained by the Vanderbilt University Medical Center. After logging into the site, we should enter the disease type. There is no type of hemangioparasitoma in the list, so let's choose melanoma, as we know that BRAF mutations play an important role also in this cancer. We'll put BRAF as the gene and the 600D as the variant. And press go. My Cancer Genome is very oriented for clinical utilities and lists only clear and documented information. For that reason, only very restricted set of diseases, genes, and variants are included, and for each, the relevant drugs are listed. After providing the details of our mutation, we get detailed scientific survey regarding the significance of the mutation to the particular disease and all relevant studies. Both in the text and in the first line in the drug table, we can see that vemurafenib is recommended for this mutation. We should remember though that all these recommendations are for melanoma, and we are dealing with a different disease here. Unfortunately, no clear evidence regarding the role of BRAF V600 variants in hemangioparasitoma exists. However, given the patient condition, the fact that BRAF is clearly involved in progression of various different cancer types, and there are approved inhibitors for this protein, it is easy choice indeed to attempt these drugs in such cases. Indeed, vemurafenib turned out to be a very effective drug in our case. In general, transforming molecular knowledge and usage of relevant respecting drugs between different cancer types is one of the main benefits and important development in the personal genomics era in cancer. I want to briefly mention other resources to obtain cancer molecular data. While COSMIC offers the most comprehensive list of somatic mutations, these mutations were collected from different sources using different technical procedures and with different qualities. Also, no raw sequencing data is available in case that complete analysis should be repeated. Several comprehensive portals offer such additional molecular data based on well-defined experimental setting and includes also important metadata regarding the samples and the patients. The GDC, Cancer Genomic Data Common, maintained by the National Cancer Institute, is such a high valuable resource. The user can analyze data within the site or download data based on samples and/or experimental procedures. Some of the data is publicly available, while some other data requires controlled access, which should be granted for the user. The ICGC, International Cancer Genome Consortium, is an equivalent international portal which offers also analysis of data based on international projects. These resources deserve much more time and space and will be covered more extensively in our future courses. Finally, so far we discussed the mutations in terms of gene names, genotype, and genomic coordinates, without addressing the actual molecular mechanisms by which they cause the functional effects. In recent years, there are attempts to allow researchers access to protein structural data in order to get basic mechanistic insights regarding the structural and functional effect of the mutations. One such tool which has been developed in the Sheba Medical Center is G2-3D, and we will explore this tool. After opening the website, we can search by both genomic information or protein information. By entering BRAF 600D, we retrieve all resolved structures in the database for this protein or homologous proteins, and we'll get indication regarding the position of the variant on the protein. Let's pick one structure with good coverage, such as 3SKCB. We can now inspect the structural context of the variant and examine its position with respect to previously reported functional variants, see its conservation and structural contact it creates with other amino acids, and how these contacts change following the mutation. Molecular functions are carried out by 3D machines, and such tools allow us to understand mechanism of action and provide starting points to address problems such as rational drug design. So as you can see, there are many available tools which help us to understand individual tumor genomes for the benefit of the patients. What we have shown here is just the tip of the iceberg, but we hope you got some idea of what can be done.